This is Lecture 4, Part 1. We're going to talk about how genes work, basically an overview of the flow of the genetic message, including mutations. This is the first of a 3-part lecture. So what we're going to talk about first is what is a gene, and then we're going to talk about what is referred to as the central dogma, including the roles of 2 cellular processes known as transcription and translation. Just to give you a heads up, a lot of students have trouble differentiating between transcription and translation. These are really important things to learn about if you want to understand how cells work. So it's really important that as you're learning, you really try to clarify whether you're talking about transcription or whether you're talking about translation, and coming up with ways of keeping those 2 sets of information separate. Both of them pertain to the synthesis of proteins and functional RNAs. They're both very important, but they are distinct cellular processes. So first, let's define a gene. A gene is a section of DNA or RNA in some viruses that encodes information for building one or more related polypeptides or functional RNA molecules, along with the regulatory sequences required for its transcription. It's quite a long definition. It's found in the definition section of your textbook, and it's very important that we move to this sort of sophisticated definition and abandon that kind of earlier high school level definition of just the blueprint for a protein. That's not really adequate for a modern understanding of how cells work. Where are genes? Genes are encoded by DNA, and the DNA is found in chromosomes. Chromosomes are made up of both DNA and protein in a structure known as chromatin. Want to remember that in eukaryotic organisms such as ourselves, our chromosomes occur in linear pairs. There's a pair shown there, and we inherited one of them from each parent, one from the mom, one from the dad. Each pair has essentially the same kind of sequences on it, so they are matching pairs in that you'll have the same gene on each chromosome. So you can imagine these yellow bands here representing a particular gene. You have 2 versions of the gene, or 2 alleles. One came from mom, one came from dad. The overall sequence will be very, very similar, but they could be different. So they could be different by 1 nucleotide, or they could be different by a few nucleotides. They encode the same gene, but they're from different sources, and there can be sequence variability. All of those chromosomes are found compacted in a structure called a nucleus. If you're talking about a eukaryotic cell, if it's a prokaryotic cell, those are the cells that belong to bacteria and archaea, there's no nucleus, and instead you actually have 1 circular chromosome. They are haploid organisms, and it's just found at a location in the cell called a nucleoid. So every single one of our cells, and we have trillions and trillions of human cells, each contain a nucleus, and each nucleus has our full genetic complement, or our full genome, which means 23 pairs of chromosomes, all occurring in matched pairs, unless you're male, in which case your XY chromosomes are different. The X chromosome is much bigger, the Y chromosome is much smaller. That's something we'll talk about more in genetics if you move on to take that course in biology. So what is the central dogma? This is a saying that we use in science to talk about how genetic information flows in a cell. So here we see a eukaryotic cell. I know it's a eukaryotic cell because the DNA is in a nucleus. It's a simplified cartoon. That DNA is there in chromosomes, and we're really focusing on just one specific short region of DNA, or a gene. So this represents a gene, okay? Not the entire chromosome. That gene is being used to make a very important molecule called messenger RNA. That's this, okay, shown in yellow. That process, as depicted by this arrow, is the process of transcription. And it's done by something called RNA polymerase. That messenger RNA is exported from the nucleus, that's what's happening here, and then it reaches an important cellular structure called the ribosome. Ribosomes are made out of protein and ribosomal RNA. The ribosome takes that messenger RNA and it basically interprets it and uses that information to make protein. What's actually coming out of there is a polypeptide, or protein primary sequence. As soon as that little green protein comes out of there, it's going to reach the watery, or it's going to be in the watery environment of the cell, and it's going to respond by beginning to fold. It's going to assume secondary and then tertiary structure. If it's a protein that's part of a quaternary complex, it'll go off and find its other polypeptides and form the final protein. But that is basically what we mean by the central dogma, or how information flows in the cell. It's the idea that DNA is used to make RNA, RNA is used to make protein. So DNA to RNA to protein.